Communications and Nature Medicine, as well as other relevant Nature journals, where again, MPJ Genomic Medicine will be part of the Nature family. And finally, we also need to continue to strive to the highest ethical standards in terms of what we publish and adhere to industry guidelines and standards in terms of input and output. MPJ Genomic Medicine must also serve authors. We have a rapid publication through our Nature editorial offices in London. We have staff that facilitate the throughput of submissions through our online system. High quality peer review, again, high ethical standards. Importantly, every original research article accepted for MPJ Genomic Medicine will have a 200 to 250 word editorial summary, which is written by a Nature science journalist. And it's meant to communicate beyond the research community the value of that work. And that's a value that we extend to authors. As you expect, we have high quality copy editing, English language editing where appropriate, and global high profile promotion through nature.com. The editorial process is independent. It is managed by the editor-in-chief, supported by a team of renowned associate editors. And again, our Nature Publishing Group staff and systems are here to support and facilitate that publishing process, ensuring a fair, efficient, and high throughput of high quality work. So introducing our journal, Dr. Al-Khattani previously presented our introduction. We announced the journal last week to an international audience. There was a press release issued both in Arabic and in English, and we've received very positive press coverage as a result of that promotion. This is the website that currently is promoting the journal. On the bottom right-hand corner, you can see the blue button, which is Submit Manuscript. So we are live and open for submissions. And I'd like now to take the opportunity to introduce Professor Stephen Shearer, who's our esteemed editor-in-chief. And I'll ask Stephen to step up and to talk a little bit about the aims and scope, and that will lead into his presentation. Stephen. Thank you.
Thank you very much, Martin. It's a great honor to be here. I think this is going to be a very memorable day, launching officially the journal. Thank you, Professor Al-Khattani, for having us. So I could say many things about the journal today. We heard a good introduction of background of what the journal will focus on. I think one of the major things we're really trying to accomplish here is to open up the area to the international community. We'd like to see as many papers coming in the general area of genomic medicine, and I'll better define that throughout my presentation later, from all countries around the world, and also, of course, from the Middle East here. In particular, because genetics is, by definition, universal, and the diseases that are affecting different populations are influenced by genetics, and we need to have much better representation to understand the applications of genetics and genomics in medicine. I think probably most of the papers that have come in the last 20 or 30 years have come from what we call Western countries. And in fact, probably the greatest impact will be in the remainder of the world. So we need to see much more activity, both in the publication realm, but also in the research realm. So one of the things we would like to do is to help actually in some of the designs of the studies in a way so that the papers are sufficiently high quality, robust in nature, and breadth so that they can withgo a very thorough peer review and be published in this journal. In some cases that may be requiring some proactive planning around study designs, and also perhaps bringing together different types of data sets and expertises. But just to emphasize, the goal here is to have a very high impact journal early on. I would actually say we go for 10. And to do that, we need to have the best papers coming forward. So we will form a network. We're putting together this week, and then in the near future, the associate editors and other editorial board members who will help write some of the papers, but also to solicit very interesting studies that are coming from around the world, and hopefully will be published in this journal in the short term. Okay. Right. So. Okay.
I'll just interrupt Steve here. Possibly some of us do not know our editor-in-chief. So first of all, I'd like to thank him for agreeing and accepting to be the editor-in-chief of the NPJ Genomic Medicine. Without his agreement and his acceptance, we wouldn't have such big success in establishing the journal. So for Steve, he holds the Galaxy Smith Kline Canadian Institutes of Health Research Chair in Genomic Science at the Hospital for Sick Children and the University of Toronto. And he is the director of Lifeline Center, if I'm pronouncing it right, as well as the Center for Applied Genomics at Sick Kids. He is also the co-director of the Sick Kids Center for Genetic Medicine. Steve has made numerous contributions to medical genetics, including mapping, sequencing, and disease gene studies of the human genome. In 2004, his team actually co-discovered global gene copy number variants, CNV, as a common form of genetic variations in human DNA. That was a great discovery. And his group then identified CNV to contribute to the etiology of autism and many other disorders. And the database of genomic variants, he found it facilitates hundreds of thousands of clinical diagnoses each year. Steve has won numerous honors. In addition that he is obviously distinguished fellow of the Canadian Institute for Advanced Research, the American Association for the Advancement of Science, and the Royal Society of Canada. In 2014, he was selected as the Thomson Reuters Citation Laureate in the field of physiology or medicine for the discovery of large-scale CMV and its association with specific diseases. So I'd like really to warmly welcome Steve as our highly cited, distinguished scientist.
Okay, thank you very much. So in my presentation, I'm going to show some of the data from our laboratory relevant to genomic medicine, and also make some comments of the need for the journal. So I've specifically selected some slides to bring the discussion back to the goals and aims of the journal. The title of my presentation is Genomics is the Medium for 21st Century Medicine. And this is based on a very famous Canadian statement back from the 1960s, Marshall McLuhan, who made the comment that the medium is the message for telecommunications going forward in the future centuries. And essentially he predicted the impact of, or the internet and the impact across society. And I would argue that we're at a stage now that technology actually is revealing the genome sequence to be really the starting point and ending point where we will tie all information in medicine back to. And I'll talk throughout my presentation now that we have the ability to decode the entire genome sequence in a single experiment relatively quickly at reasonable costs now. So in addition to understanding the genome sequences of our own species and comparing in a comparative intra-species genomic comparisons, we also can compare the genomes to other species and look for conserved sequences and specific regulatory elements, for example, but also develop, based on genome-wide data, new hypotheses that spin out by looking just at the entire genomic sequence data. And I think this is really an important conceptual change of how we're going to be doing medical research into the future. And I'll come back to this throughout my presentation, where we actually will sequence the genome probably first, even before there may be a clinical presentation, and not only predict what the clinical outcomes may be, but by comparisons of sequences to family members and also other members in the population, identify new risk factors just based on sequence information itself. So what is genomic medicine? This is a slide that was given to me. I actually don't know where it's from. I checked on the internet. I could try to acknowledge it, but I couldn't find it anywhere. It was given me to a medical geneticist in our department. And I think when you go see a doctor, they will always ask you to stick out your tongue and say, ah. And what we think is happening now, as I'll show you in a moment, is you'll do the same. The doctor will do a physical examination, but at the same time take a swab of saliva or perhaps a blood sample and determine a genome sequence. And genome sequencing will actually become a surveillance mechanism in the medical workup, not only of individuals who are coming to the clinic or the hospital, but actually of the general population, to the healthy population. And what I've added here is a comment that I presented to our own hospital. I work at a pediatric hospital in Toronto, the Hospital for Sick Children. We, in a way, learn quite a bit from publications from different journals coming from Europe and the United States quite often. And when I was presenting this to our board and developing our own genomic medicine center, I said we really need to roll up our sleeves and do this work here in our own pediatric hospital, because the lessons that we're learning are very specific to our own population. Our healthcare system is socialized healthcare. We study primarily pediatric diseases, but quite often we're studying diseases that go from pediatrics into adolescence and adulthood. So we actually have to translate the information across systems. And in Toronto, we're a very multinational city. In fact, apparently it's the most multinational, multicultural city in the world now, even beating London and New York City. We have 200,000 new immigrants coming each year, and the biggest challenge we have in the diagnostic laboratory are new diseases specific to these populations that actually come to the clinics. So we need to understand their genetics, and I think this is a huge area for growth and collaboration around the world, and this can be enabled by the new journal. And I'll give you an example in a moment. So much of genomic medicine will be driven by technologies, and the three predominant ones are shown here. Of course, karyotyping is still the number one diagnostic test that's performed in North America, and it's probably the case here too, but that's rapidly changing. So for over 50 years, karyotyping has been used in, of course, congenital abnormalities and cancer workup. And in the mid-2000s, million feature microarrays started to take foothold. And in our laboratory now, in our diagnostic lab for most developmental abnormalities, microarray has now supplanted karyotyping as the primary screening technology. And then now, more recently, we can actually do exome sequencing, so to sequence the 1.6% of the genome that encodes genes, or do entirely whole genome sequencing. And I'll give you some examples a little bit later. But the point I wanted to make here is that these are the DNA-based technologies. We're really moving into now whole genome testing. Okay, single gene sequencing, of course, is very important for specific disorders, but I think to really understand complex disease, even monogenic diseases like cystic fibrosis, you still need to have the whole genome sequence or whole genome assays. And typically after the introduction of a new technology in the research laboratory, it takes about, in our experience, 5 to 7 years to see that technology take place in the diagnostic laboratories. So while we're now doing whole genome sequencing in the thousands in our genome research center, the diagnostic laboratory has not yet introduced these technologies, but that will come in a few years' time. So what we see here is a slide where we've combined all of the data from our hospital, and all of the diagnostic testing is actually done in hospitals in Canada, and Sick Kids Hospital has the largest diagnostic laboratory. So we've combined the whole genome assays shown in different colors here. So clinical microarrays, research microarrays based on RNA, exome sequencing, and now whole genome sequencing. And you can see the shift from doing single gene-based or locus-based testing towards whole genome assays over the last few years. And this has really been, I think, the major change that we've seen. And the advances that have come are the ability to do, again, in a single experiment, and assay the entire complement of genes or gene products, and now whole genome sequencing. So I think you're probably also experiencing that here, perhaps to a lesser extent, because we've had such a push in the technology development and translational aspects. But I think the journal will capture all different aspects of this. So not only the data generation, but the technology, development aspects, how the data is translating into clinical application in the laboratory, and then how it actually affects the populations that these technologies are serving. Because how they're implemented in Saudi Arabia will probably be much different than how they're implemented in London or New York City or Toronto. One of the things my laboratory has studied over the years is a simple, very simple question. When I was a student, I always wondered how much genetic variation is there in a genome? We still don't actually know, believe it or not, especially with this garbled slide. But if you look, this is Craig Venter's genome, and I've had a PhD student who just graduated who's been studying this since 2007. And we've thrown every different technology at Venter's genome. Of course, his genome was the one that was sequenced in the Celera Human Genome Assembly, which was Sanger sequenced. We've run the genome on every different microarray and every different next generation sequencing technology. And what I wanted to point out here is that we know quite a bit about the single nucleotide changes, 3.2 million roughly per genome. And we're learning more and more about the copy number variants and the insertion deletions. So we see roughly 40.5 megabases, so 10 times the number of nucleotide bases affected by unbalanced changes, so copy number variants, compared to the SNP type changes. And if you look at the slide here, if you have really good eyes, you'll see that the distribution of variants is shown based on the size of the variant on the x-axis, and then the number of variants detected in Venter's genome on the y-axis for each of these different categories. And there's a little bit of an increase in variants in the 10,000 range, and the reason for that actually is in the Sanger technology that they used, they used paired end libraries that were primarily 10 kb in size, so they actually have a better yield of variants in that size range. So I expect that when we have even more complete sequencing of this genome and other genomes, we're going to see an increase of each of these different bars. Okay, so it's still not a perfect genome sequence yet. This is the best genome you'll find. But what's most important is that roughly 20% of the protein coding genes are impacted by a copy number variant or an indel in the heterozygous state in some way or another. This was really, really shocking. Of course, the major message from the Human Genome Project was that we're 99.9% identical. And this set of studies really showed that that's not in fact the case. So there's a significant amount of genetic variation in the genome that we really have to understand better from worldwide populations if we're going to apply genomics in medicine. This is the database that was mentioned that we host. We've been running this since our 2004 paper, cataloging from all of the major studies, all of the CNVs and indel type data. We work with the EBI in the UK and the NCBI. We curate all the data in Toronto, and then it goes into these other databases, for example, the UCSC browser. And even to date, even though it's been 10 years now, there's still only 22 well-conducted studies that capture CNV from worldwide populations. Okay, and still, again, thank you. Can you open it for me? Yeah. It's mainly from Western populations. So this is a real challenge. So one of the areas that we'd like to develop also through the database, through the paper, the journal, is a resource section. So if there's some very specific and needed resources, say in data cohorts, for example, or types of data that might help better populate this type of database or others, we'd like to entertain publishing those. These are very high impact papers. This paper we published in Nucleic Acids describing the database. When was it? Just came out, I think it was last year. It's already been cited over 100 times. So these resources really are hugely valued and hugely utilized. So this is something we may be looking forward to. And this is the reason why. There were 3 or 4 papers published in clinical journals that were using microarrays to scan intellectual disability in autistic patients from around the world. And 3 of the 4 papers actually it implicated this locus, the YWHAE locus, as being pathogenic in intellectual disability. And one of my students was looking at this, and we saw the same observations in our clinical cohort. And I asked the question, so, what populations is this CNV deletion coming up? And this was a heterozygous deletion. And because we had the population data underlying the clinical screening that was being done, we could see actually in the 2 cases we had identified, which are shown at the top, they were from Asian background. And then we were able to go back and look in the literature, and all of the papers that were published were from a heterogeneous population. Whenever they saw this deletion, it was actually from an Asian individual. So in fact, it was turns out, kind of long story short, and we published this now, this is a common polymorphism in the Asian populations in about 2% frequency. We don't see it really in the non-Asian populations, and it's been misinterpreted as being pathogenic in clinical microarray studies in many clinics now. So since we published this, they have to go back and actually reinterpret the data, call back the families, and counsel them accordingly. So again, this is the kind of study that I think would have very, very important specificity in different regions of the world, such as the Middle East. This was a paper we just published, it came out last week. I was on the cover of Nature Reviews Genetics. And what we did here is we did a 10-year meta-analysis of all the data that's in the database for genomic variants. So we thought long and hard about this. We have an advisory board of many people's names you would recognize. And they said, we really need to have a gold standard copy number variation map of the genome. It doesn't exist at the time. And they said, your lab's the only one in the world that could probably do this because of your expertise. And you have the data, and you can actually do all the computational analyses. So we set out and did a meta-analysis. It took us 2 years to actually do this analysis. And the primary, the hardest part of the analysis was actually to identify those papers that had the highest quality data. Of the 22 that were in the database, I think in the end we used about 16 of the papers and went through some very rigorous comparisons. We're able to eliminate some of the papers that probably had a lot of false positives and false negatives, and ultimately came up with this very high quality data set and then developed a CNV map of the genome. So this gold standard data set is now being used in many clinical laboratories around the world, even though it's actually only been developed now for a few months. And also it's going to be the gold standard reference in the database for genomic variants and other databases too. So we think it will have incredible impact. We had to negotiate with Nature Reviews to do this. They publish apparently 1 paper a year where they allow you to do an analysis paper. Most of the papers in Nature Reviews are reviews. But they had done one on microarrays a few years back. We talked to a few of the other Nature journals, and they actually didn't want to publish this. But Nature Reviews said they would look at it if we did the analysis, and eventually it was published. And they put it on the cover. It's a beautiful cover design. So again, this is the kind of paper, if you come to us with an idea that we think will have big impact and you're willing to go through the proper study design, I think this is something that we need to look at seriously. So I also wanted to say, when we first published the 2004 CNV paper, and then later we published the first map in 2006 in Nature, we predicted that the amount of copy number variation in the human genome across populations would be about 12%. We were heavily criticized at that point. In fact, there was a News and Views where someone wrote, I won't mention the name, said this is bullshit. I mean, really, sorry. and then, so anyways, at the end of the day, when we redid this meta-analysis using different levels of stringency, we got 12%. So we were quite happy, actually, that our prediction was right 10 years later. This is our center at the Sick Kids Hospital in Toronto. It's very similar to CGMR here. We have different core facilities. We have about 100 staff working in these different facilities. And we're primarily doing now sequencing, and I think we have the biggest microarray laboratory probably in North America now, because we're doing population-based studies. We're doing a lot of population studies where we need to have low costs and high throughput. Sequencing is not there yet, so we're doing microarrays. But we're now doing sequencing. We have, I think, 20 next-generation sequencing machines, and most of our new hire staff are actually informatics scientists. 40% of our staff are informatics scientists. We launched the Canadian Personal Genome Project in 2012, and it was launched to much fanfare, and the Globe and Mail is the equivalent of the New York Times in Canada. It's kind of our national newspaper, if you will. And the idea here was all of our experiences in the past had been sequencing mainly pediatric cases that had specific diseases, and we wanted to see how the technology was actually going to influence the adult healthy population. So we wanted to develop consents that were specific to our own population, figure out how we're going to actually develop algorithms to decode the data, and then resources based on this, this new technology. So cut a long story short, at the launch of this and in the papers, we had 6,000 people who wanted to actually have their genome sequenced. We prioritized those who could actually pay for it themselves. We work with a private healthcare clinic in Toronto. There's only one. As I said, healthcare is social in Canada, but we have one private healthcare clinic. And then we did genetic counseling. All these, you have to pass a test to be enrolled in the project. And we've now done the first 20 genome sequences of the Canadian population. Okay, so I haven't told Martin this yet, but we're hoping that maybe we'll publish this as our first paper in the journal. We've developed a new pipeline to do whole genome annotation. No one's done a more thorough annotation of any genome as we've done here. And we've translated all the data back to the healthy individuals, and they're not that healthy when you look at their genome through genetic counselors. So we have all these lessons learned in this study. So we think that we'd like to see other papers like this coming from different regions of the world, because you're going to interpret the data based on the systems that you're operating in. We've also used this as a resource. Of the first 20 individuals, we've developed induced pluripotent stem cell lines of each of these individuals, and we're developing that as a bioresource. Using CRISPR-based technologies, you can introduce lots of different mutations and then use those to overlay combinations of mutations to do functional testing. So we're tying all this data together. So again, coming back to the resource idea, perhaps we'll be publishing papers of IPS lines, for example, that have been characterized in different diseases. It's something to think about. Just to say that the technology, I think when we look back 10 years from now, that 2015 will be the year, the inflection point where genome sequencing really takes the stage. Our laboratory, as you'll see at the end of my talk, is doing 10,000 genome sequences this year. There's lots of other large projects ongoing. The price is in the $1,500 range now for genome sequencing and using the new Illumina system. You'll see lots of these machines coming out in 2015. So I think this is going to be the year where sequencing really takes foothold. I'm going to go through a little bit quicker. I don't know how much time I actually have. But we also have a large endowment, and I think this is relevant because of some of the people in the audience. In Canada, there's a gentleman passed away 50 years ago. His name was General McLaughlin, and he was the equivalent of Henry Ford in Canada. He started the General Motors of Canada. When he passed away, he left the largest endowment in Canadian history to our university, and that was now directed to genomic medicine. Okay, so I'm the director of that center. There's, I think, 7 teaching hospitals in Toronto affiliated with the University Medical School. So we're funding specific projects doing this type of sequence-based analysis of different diseases and how we use that data. I won't go into all the different approaches, but it's going from pediatric hospitals to adult hospitals to specific hospitals that deal with, for example, women's health issues and cancer, for example. But the project that we've used these technologies on most because of the importance to the populations that our hospital serves, and then, of course, to my academic research group, has been autism spectrum disorders. And this is a behavioral condition that we now know is founded in biology. We know genetics has a significant component. And I'm just going to make a few comments of our progress that all relate back to the ability to scan the entire genome. Okay, just make 5 points here why this research is so important. The truth will be found in science. For over 30 years, the blame for autism was actually put on parents for not raising their children properly. Because it was not known that it was actually a biological condition. It was actually considered to be strictly a behavioral or an environmental condition. We know that earlier detection of autism and related conditions, because there's many different syndromes that can overlap, can benefit the health outcomes. So the earlier you can diagnose it, the better outcomes that children have. So this is a real application of genomic medicine. Better understanding of autism will benefit medical management. I'll show you some examples a little bit later. There's no drug treatments for the core features involved in autism at the present time, but there's lots of medical complications like epilepsy, for example, seizures, that can benefit specific drugs, but you need to take the right drugs. You need to take them at the right times. We're already showing applications. And of course, the discoveries are opening up new pathways to develop new drugs to the core features involved in autism. So what is autism? It's a heterogeneous spectrum disorder involving impairments in 3 domains of function: social communication deficits, restrictive repetitive behaviors, and language deficits. I think on my next slide I have, but it's now affecting roughly 1 in 68 children in North America will have a diagnosis of autism or autism spectrum disorder. And these statistics are uniform worldwide. Okay, there's been studies now out of South Korea, Europe, Canada, United States. I don't know that there's been a study out of Saudi Arabia yet, but I think there's some ongoing that show that roughly it's 1% of the population frequency. Highly genetic, there's a 50 to 90% concordance rate in monozygotic twins, and this drops to roughly 10% in dizygotic twins, which has strongly suggested that genes are involved, and of course we've now identified some of these. 18% risk to siblings compared to the 1% general population risk. So genetics plays a significant role here. The 4-to-1 male to female ratio suggests that, of course, X chromosome may be involved. And probably unknown to most is 20% of parents who have an autistic child are also on the spectrum. So there's inherited variants that can be involved. The real conundrum or enigma here is that the significant variable expressivity or clinical presentation in the same family. So you can have identical twins who have the same genomes, and one child can be severely autistic and the identical twin could be actually high-functioning Asperger's syndrome. That occurs in about 10% of families. Usually they're more closely related in their clinical presentation. There's been a decade now of genome scanning studies, primarily looking at SNP variants, and there's been no SNP that has approached genome-wide significance and been replicated. So it's thought that common variants have a minor role, although that's still being teased out. And the story, at least so far, is that rare variants and genes and mutations, rare mutations are really the most impactful in ASD. And to cut a long story short, this is what we now know. Autism presents in a form of related syndromes in roughly 10% of cases. Things like Fragile X syndrome, tuberous sclerosis, PKU. There's over 108 different syndromes that can have an autistic presentation. Okay, so having the diagnosis with respect to autism and all the other clinical manifestations is really important to make sure that children are going to the right clinics. If you screen using karyotyping, you see chromosome abnormalities in roughly 3 to 5% of cases, depending on the cohort that's examined. Using microarrays, going back to the technologies, you see copy number variants, either de novo or rare inherited, in roughly 5% of cases. That goes up to 10% if you look at higher resolution arrays. And if you use exome sequencing, you add another 5% yield of de novo variants affecting genes that are now known bona fide ASD genes. There's over 100 or so now autism risk genes that have been identified. And all of them are actually implicated in some way in intellectual disability. This is a very important point to keep in mind. So my slides are a little bit distorted here, but I think the message still comes across. There now are diagnostic tests available for autism, and these are all based on genome-wide microarrays right now. And the top 5 loci that come out in our Canadian cohort are shown on the left-hand the slide here. We've done a meta-analysis of our data plus other publications showing these loci. The take-home message here that all of these, even though they're significantly associated with autism, they're present in less than 1% of the population. Okay, so these are rare mutations. And then perhaps even more importantly, we're seeing the same genes come up, or the same CNVs come up in other neuropsychiatric disorders. So the same 16p11.2 deletions we see in autism, we see in schizophrenia or bipolar disorder or ADHD, and intellectual disability. So we've now moved to doing whole genome sequencing. And the reason for this is there's so many different genes involved, we need to actually capture the variation in combinations of genes. I didn't have time to talk about it, but if you do the CNV studies and identify the loci and compare those to what exome find, there are different classes of genes that are coming up. It's because we think the different mutations are contributing in different ways, different mechanisms. So we want to capture all the variation in one experiment. So again, we need to do whole genome sequencing. If you do exome sequencing alone, the capture arrays that are presently available don't capture all the exons in the genome. And if you look at the known ASD genes, at least 9% of the exons that could be affected are actually not represented. So again, you need to sequence the whole genome. And then in addition, there's a lot of published literature, including our own, that have shown that intergenic mutations or intronic mutations are well characterized to be involved in autism. So again, you can't just look at the genes, you need to look at the whole genome. So we published this paper in 2013 and another one that came out just a few weeks ago, where we did whole genome sequencing on 32 families and could identify mutations in 50% of those families, so 16 out of 32, where the data would have implications for the medical management or genetic counseling of the families. Okay, so this essentially doubled or tripled the yield compared to microarray and exome sequencing. I won't go into all the details, but I could tell you a story of each of these families that are shown here and how it impacted the management of their children or the genetic counseling of future children. This is just one example here where we identified a de novo loss of function mutation in the vasoactive inhibitor gene, VIP. This was interesting because, in fact, we thought it was a de novo mutation. It would be unique to the development of the male index case shown by the arrow on the left-hand side here. But in fact, when we looked at the segregation in the families, we did not see it in either the parents in their blood DNA, but we found the mutation in what was at the time an unaffected brother, yeah. And then we went back and actually looked to see, because we had collected this DNA a few years back, it turned out that this brother is actually on the autism spectrum too. So had we had that information much earlier on, had we had the molecular data much earlier on, we actually would have been able to counsel this family and get these children into earlier behavioral intervention therapy. So we think this is actually a somatic mutation in the germline, probably of the father. And then this is another example of the paper we just published in Nature Medicine, where we used, again, whole genome sequencing technology, but we looked at families that had 2 known siblings that had autism. In the previous case, we actually didn't know the second child had autism until we went back and looked when we had the molecular diagnosis. And I just picked, again, in 40%, it was 45% of the families, we had information that went back to the families using the whole genome sequencing. And I've just highlighted a few examples here showing the complexity. So in sample A, family A, we identified, again, a de novo mutation that we thought was de novo, but it turned out to be a germline mutation in these 2 affected sisters. And this was a deletion of the SCN2A gene, which is it was a known epilepsy gene for many years, and now it's turned out to be one of the predominant autism genes that's been identified. And this mutation was undetected using microarrays and exome sequencing because it was a small deletion that occurred in the introns flanking exon 18 shown on the right-hand part of the slide there. So we didn't have the sensitivity to actually detect it using these other technologies. Family C is an unrelated family where we found different types of mutations. We found a mutation in the index case on the left in the syntaxin XBP1 binding protein 1 gene. This is a de novo mutation. This was also an epilepsy gene. Now it's being found in autism now that we've been looking closer. We think it's actually causing the autism in the female. And then the affected male has a frame shift mutation in the UBE3A gene. This is an inherited mutation from mother. You know, this is actually the Angelman syndrome gene. and it's imprinted so the female carriers are unaffected. And we think that this is actually the causative mutation. Angelman syndrome, 40% of the kids have autism. So in this case, had we had that information at the time, these two children are actually in their 30s now. They're not children anymore, they're adults. We collected these DNA samples 25 years ago. These individuals would have been seen in completely different clinics and treated in different ways. And then on the right-hand side, we see a mutation, a de novo mutation in the thyroid hormone receptor A gene. And this was a new gene when we had actually first found this when we did the sequencing about a year ago. And since that time, 2 other papers came out from other populations in clinical journals that showed mutations in intellectual disability. One actually was in an autistic individual. So again, having this data out in the literature was critically important for us to make the association of our de novo variant. And then in the affected sibling, we found an inherited mutation being passed from mom to a male boy in the CATNL2 gene. Again, this is a gene that's been coming up in other studies to be an autism susceptibility gene. This is the first time we've seen it inherited. We have to go back and look at mom to see if she may be on the spectrum, but females tend to be more protected, so we'll have to wait and see. The take-home message from the study is even in siblings in the same family, same biological family, when we had the information available, 2/3 of the sibling pairs carry different rare variant mutations. Okay, kind of playing back into the importance of being able to assess the entire genome doing this type of interpretive experiment. So I'll just close by our latest project, and I think this may take us into where the future of genomics is going. We've now determined, of course, the significant genetic heterogeneity in autism, and we've estimated that we have to probably sequence well over 10,000 families, whole genome sequencing. We've initiated this project with Autism Speaks. This is the CSO of Autism Speaks, Rob Ring. He came to visit Toronto. They're funding this, and we've developed a collaboration with Google in California where we're putting all of the whole genome sequencing into a cloud database as it's being generated. We've now completed, the Nature Medicine paper that came out a few weeks ago, we put the first 1,000 genomes into the cloud database. We have 2,000 more going in the next month or so, and then we think we'll have 10,000 done by the end of 2015, we hope. And the idea is that we want to bring as many different creativities and minds and eyes to the data as possible. We think we need to have computer scientists involved, people who are doing machine learning research, pattern finding, and we think this is going to take an international effort, right? So once we have the data out there, we can collaborate. People can go take their tools they develop into the cloud to do their experiments. They can download the data if they need to. We'd rather have them do it in the Google Cloud because it's probably more efficient that way. But it's there for anyone to use in the same way you would use the internet. We really want to take open science to its extreme. So we're hoping that if other whole genome sequencing experiments are being done in autism, say in Saudi Arabia or in Asia, wherever it may be, or even in related conditions that have an autism phenotype, intellectual disability, or epilepsy, we're going to put all this data in the cloud. Autism Speaks is going to pay for all of this so that people here can sit on their computers and it doesn't cost you anything other than to do your experiments and have good ideas. And I think this is really where science is going. So when we're thinking about the development of the journal, we may publish perhaps some papers on the type of tools one would use in this type of interpretation, for example, analyzing the data itself, translating it into clinical applications. And I think with that, I will close and say thank you very much.
Thank you very much, Steve. I think from Steve's talk, you realize what sort of level that we have as it achieved for MBJ.